But for those of you that have, what is the average amount of blood volume that an average person has?
5 to 6 liters.
5 liters. 5 liters of blood. And if we think of the percentage of our body's mass, our blood makes up what percentage of our body's mass? Is it a lot? No, our bones and our muscle is really what makes up most of our mass. Even though we have 5 liters of blood, it's not making most of our mass. It's really only about 8% total body mass. But it's really important. All right? Now, if we broke up blood into its different components, first of all, what type of tissue is blood?
Connective.
Connective tissue. And connective tissue has an extracellular matrix, right? What's the extracellular matrix of blood? This is in addition to the cells that make up blood. So what's not a cell when we think of blood? I think somebody said it. Fibrin is going to be a solute in this component of blood.
Somebody did say it. Plasma.
Plasma, yes. Plasma is the extracellular matrix. This is the extracellular matrix. Because blood is a connective tissue. And somebody said fibrin. But the biggest component of plasma when we're talking about proteins is what? Albumin. And so you're going to have albumin. You're going to have electrolytes in here. You're going to have antibodies. You're going to have hormones, glucose. And all these things are going to be dissolved in what?
Water.
What?
Water?
Water. So water, then, is the solute or solvent of this plasma solution? Solvent. These guys are all wet. They're dissolved in that solvent. We call things that are dissolved in a solvent what? Solutes. Plasma makes up about 55% of whole blood. So this is in addition to the cells. When we talk about the cells that make up blood, we call them formed elements. And formed elements are either cells or fragments of cells. What am I talking about when I say fragments of cells? Platelets. Platelets are going to be involved in what process? What kind of function do they have?
I know they come into play during coagulation.
Coagulation and blood clotting to stop bleeding. Exactly. So platelets are dealing with blood clotting. And that's what we're gonna talk about that on Thursday.
I have a question.
Yeah.
Why aren't antibodies considered a formed element?
Because that's a protein. Okay, so it's a solute. These are cells or fragments of cells. Other questions? Good? Okay, cool. What's another cell that, just a general name of it though? What are the ones that have an immune response? That's their function. What are those types of cells?
White blood cells?
Yes, leukocytes. These have immune response. And then we have our erythrocytes. And these are transporting oxygen to tissues. Eek to tissues. And this is about 45% of whole blood. So if you add 45 and 55, that's like 100%. So these two here are these are less than 1% of your total whole blood. All right? Now, we're going to see that to make these formed elements, and in particular leukocytes and erythrocytes, what is it called when you make formed elements? What is that process called? When you make red blood cells, it's called erythropoiesis. But if you make any blood cell, it's called what? Hematopoiesis. So that's the general term for making of blood cells, whether it's leukocytes or erythrocytes. When we're making erythrocytes, it's erythropoiesis. Now, we're going to see that, and so now I'm going to really focus on these erythrocytes, because that's what this chapter's about, is the red blood cells. And a mature erythrocyte, does it have organelles? No. Instead, what is it mainly composed of? Hemoglobin, right? So a mature red blood cell is essentially a packet of hemoglobin. Right? And when we're talking about a red blood cell, is it a sphere or does it have some kind of funky shape to it? It's a biconcave disc. So if we think about something like a sphere, so here's a sphere versus something that's shaped like this, that's packed full of hemoglobin, the ratio of surface area to the hemoglobin in that cell, is it higher here or here? Like, where do you have a higher surface area compared to the hemoglobin within it? Do you have a higher surface area to hemoglobin content in just a sphere or in a biconcave disc?
Yes.
Biconcave disc, right? Why is that important? Why is it important to have more surface area in a red blood cell?
Diffusion of gases.
The diffusion of gases, in particular the diffusion of which gas here? Oxygen. So if you have a bunch of hemoglobin here, oxygen's in the middle, it's going to take longer for it to get out of that cell. Where if it's here, boom, it's out. And that's what we want. We want to be able to deliver this oxygen quickly to the tissues so they can function properly. So this is bad news. This is what it is. And this is like a side view. If we were going to do it like this, they always do this kind of thing to show it's biconcave, the little smiley cell. Now, hemoglobin is hemoglobin a tertiary or quaternary protein?
Quaternary.
Quaternary, why?
So it has 4 binding sites for
It has 4 of what?
heme groups.
Okay, heme is going to be this molecule that has iron that's going to bind to oxygen, but we're talking about a protein. And so proteins, when it's quaternary, are made out of subunits. So it has 4 subunits that are called globins, all right? And it's going to have, and I'm only talking about adult hemoglobin. Fetal hemoglobin is different, but you have 2 alpha globins, and you have 2 beta globins that make up hemoglobin. So it has a quaternary structure. Now, each one of these globins has what Angel's talking about, a heme group. So each globin has a heme group. All right? And this heme group is made up of this protoporphyrin. I don't know if I'm spelling this right. Okay, still spell it wrong. Oh, there's an I here. Something like that. Protoporphyrin ring is just like a chemical ring structure, all right? That's going to be this sort of disc in each one of these globulins. And attached to that is going to be iron, all right? And that'll make end and iron. Now when we're talking about the iron with this heme group, you'll have Fe2+, it could be Fe2+, or it could be Fe3+. Which one of these is reduced iron? Let's think about what reduced means. Gain of electrons is reduction, that's the GER, and then loss of electrons is oxidation. Now, are we going to have a more positive or a less positive if we have more electrons?
Less positive.
Less positive. So which one is the reduced? Fe2+. This is reduced. So whenever this heme has Fe2+, we're talking about reduced hemoglobin. We're not talking about number of hemoglobin. So when you read that in your book and it says reduced hemoglobin content, they're referring to hemoglobin that has Fe2+ with its heme group. This guy binds to oxygen. When you get oxidized iron, it will not bind to oxygen. That's going to limit the ability of that hemoglobin to bind to oxygen. It's just not going to do it. Okay, but we want hemoglobin to bind to oxygen, all right? So the oxidized form of iron does not oxygen. And that can be really bad because oxygen is not very soluble in water. The way that oxygen gets to the tissues is by binding to iron within a heme group, okay? Everybody good with that? Okay, cool. Now, if our hemoglobin has 2 alpha, 2 beta, and each one has a heme group with reduced iron that's binding to oxygen, how many oxygen will be binding to one hemoglobin molecule? 4, right? Because every single one of those globins has a heme group, and there's 4 globins, okay? So you have 4 oxygen binding to each hemoglobin protein. And I think there's like millions of these hemoglobin proteins in these red blood cells, in one single red blood cell, right? So let's look at what's happening to make this hemoglobin, all right?
I have a question. So
Yeah. Do I keep this up? Can I read it?
Yeah.
What's your question?
So the heme group is made of the protoporphyrin ring and iron?
Yes. Any other questions? All right, now, let's say you have a red blood cell. It's not mature yet, okay? So in this process, we still have organelles in it. We still have ribosomes in it because we're making what? What is an immature red blood cell doing? What's its primary goal?
Building hemoglobin.
Making hemoglobin. That's its primary goal because in the end, all it is, for the most part, is a bunch of hemoglobin inside this cell. Nothing else. Not a nucleus, not any organelles, no ribosomes. It's predominantly just hemoglobin. So we're going to see that when this immature, this is an immature red blood cell. Key players is going to be mitochondria and ribosomes. Ribosomes are going to be really making what component of hemoglobin? What do ribosomes do? Yes, they are going to, with RNA, translate that RNA into proteins. That's going to happen here. So the ribosomes are going to make the alpha and beta globins. So what do you think the big thing is with mitochondria? What is it? It does make ATP, but in regards to making a hemoglobin molecule, what is it going to make? What component of that hemoglobin molecule? We got the globins. What don't we have yet? The heme group. Mitochondria is going to make the heme group. Okay? And then these two are going to combine and make hemoglobin. Now, we're going to see that the mitochondria, to make a functional heme group, is going to have to get something from the diet. Okay? And so what is needed from the diet to really make a functional heme group here? Yes. So we're going to see that you're going to have this protein outside of this cell that's going to bind to another protein that has iron. Okay? This protein that's bound to iron is going to be called transferritin. And think of it as the protein that's going to, what does trans mean again? Transverse section of the body is going length of the body. It's a transverse section, it goes across the body, right? Transferritin protein is going to go across the cell membrane. And it's going to enter into the cell through phagocytosis. Then this iron can go two places. It can go into the mitochondria and become a part of the heme group, and some of it will bind with a protein inside this red blood cell and be stored. And when it binds to this protein is called ferritin. So if you look at the actual symbol for iron, Fe, ferritin, the protein that stores it, Fe, ferritin, right? The thing that moves it across the cell membrane, transferritin, all right? And that's how you make hemoglobin, all right? Pretty good with that? Cool. Now, if we're talking about all the things that we need, yeah, we need iron, right? But what else do we need to make hemoglobin? What do you need to have in your diet to successfully make hemoglobin? Minerals, correct? You need some minerals, right? You need some minerals here. Maybe protein in your diet? Protein and amino acids. Amino acids. The other thing that's really important in making hemoglobin is B vitamins. And in particular, B12 and folate, which is a type of B vitamin, they're important for mitosis to occur, for it to divide, all right? If you don't have B12 or folate, do you think that a mature cell would be bigger in size or smaller in size? It wasn't able to divide. So it's going to be bigger, right? Because it wasn't able to divide. And so when you have these types of anemias, B12 and folate, you're going to have what's called macrocytic cells because it wasn't able to divide. All right? So these will result in macrocytic cells. Now, in regards to iron, you're not able to make this heme group. It can still divide though. But now you're going to have essentially a cell that isn't going to have as much material in it because this heme group isn't being made. So these are going to be, when you have lack of iron, microcytic. Everybody get that? It can divide stuff. So these will be smaller because of that. Now, when you think of iron, and you think of like when iron oxidizes, you get rust, right? So when you look at your red blood cells and that iron is binding to oxygen, you have this really bright red appearance to them. But if there is not even any iron with that heme group, those cells actually look very pale. They don't even look red at all because they don't have iron. And so they're called hypochromic, meaning they don't have that reddish appearance because they're lacking iron. So when you don't have enough iron to make hemoglobin, the cells are going to be hypochromic. All right?
What is, how does a cell become microcytic again?
It's because the hemoglobin within that cell, it's not making the heme group, and you're not making as much hemoglobin, functional hemoglobin, so the cell is smaller because of that. It just has less in it. Other questions? All right, so this is how you make the hemoglobin. Here's your immature red blood cell. Once you've made all this hemoglobin, the ribosomes start to degrade, break down. Mitochondria gets kicked out. Nucleus gets kicked out. And then you just get this mature cell that's just packed full of hemoglobin for the most part. Now, if we're talking about fetal hemoglobin, the difference there in fetal hemoglobin is that instead of beta chains, fetal hemoglobin, I'm just going to abbreviate hemoglobin HB. It'll still have 2 alpha chains, or globins, but instead of 2 beta, it has gamma globins. I really meant to put globins there, but anywho. Now, the cool thing about fetal hemoglobin is that these gamma globulins have a higher, even a higher affinity for oxygen than the beta globins. And that's important as that fetus is growing. It needs to be able to have that hemoglobin really stick to oxygen, so once it gets to the tissues that are metabolically active, it has a lot to distribute to those tissues. About 6 months after a baby's been born, these have been converted to beta. So this is short-lived.
Some Andean populations, they have more
Oh,
gamma
really?
as adults. It's more efficient at carrying oxygen.
That's really interesting. It's
Yeah.
like a reversion.
Yeah, I'm half Bolivian, flying into that airport, and then you fly in at 16,700 feet, and so they've adapted.
Those genes get turned on to start making gamma globins to account for that low oxygen in their air. Hey, that's cool, thanks. Sweet. I can erase this. Now, think of a mature blood cell. Its primary activity is to deliver oxygen. But it still has a cell membrane, right? It still has proteins that are a part of that cell membrane, right? Do you think it's going to need to make ATP to maintain its structure? How is it going to do that? Is it going to do it with cellular respiration? Does it have mitochondria, a mature erythrocyte? No, it doesn't. So, a mature erythrocyte still has to make ATP, but it doesn't have any mitochondria. Erythrocyte. Still has to make ATP, right? So it still needs ATP, but it doesn't have mitochondria. What is it going to do to make ATP? Does it have to make a lot of ATP? I mean, it already made all that hemoglobin, right? It's only going to survive for 120 days, and then it's going to die. But it still has to have ATP. So what process has to happen before cellular respiration? Before you, that also makes, 2 ATP, net ATP. What is that? Glycolysis. So the mature erythrocyte is going to do glycolysis. And when we're talking about this process, glycolysis, they give it this name. Embden-Meyerhof pathway. Because actually glycolysis, it has all these intermediate steps before you get to pyruvate, right? And some actual steps in glycolysis can be different. But the one that typically occurs in our cells is this Embden-Meyerhof pathway, okay? And so that's how a mature erythrocyte gets its ATP. And it's really important to make this ATP in particular so that you can regulate intracellular ionic concentrations. What is one of the most famous pumps that helps regulate ionic concentrations inside of a cell? What is that pump that makes sure you have more sodium outside than inside and more potassium inside than outside? What's that pump? Sodium-potassium pump, right? So if you aren't making ATP, that means your sodium-potassium pump isn't working, and what's going to happen to that cell? What's the thing that happens in just cellular injury? What's the very first thing that happens because the pump isn't working properly? Hydropic swelling, exactly. So if, just like in everything else that we've learned, if this erythrocyte cannot do glycolysis with this pathway, and it can't regulate ionic concentrations, then that means your sodium-potassium pump doesn't function. So if can't make ATP, sodium-potassium pump does not function, which leads to hydropic swelling. All right? Which is not good. And we start to go from that nice biconcave cell to a spear now that isn't doing its job properly. It's not functional. The other thing that's going to be produced in this pathway is this molecule called glutathione. I might have that spelled wrong. But this is just an intermediate within all those steps in glycolysis. And what this guy does is it actually is going to react to oxidants. So it's sort of like an antioxidant here. those oxidants, we want to neutralize them so that they don't start disrupting that cell membrane. So that's what this Ebden-Meyerhoff pathway is doing. Everybody good with that?
Can you repeat that again? For the-
Okay, so another intermediate, because we got to realize that it doesn't go glucose to pyruvate right away, right? You're going to have the transfer of phosphates from one molecule to another. You have something like 10 steps overall from going from a glucose molecule to pyruvate. And one of the intermediate things in this is this molecule that acts as an antioxidant. Okay? Yeah.
Would it do anybody any good if they took a glutathione supplement?
Huh. Maybe. I mean, it's going to react with those oxidants, so that could help with, like, you know, people say, hey, eat blueberries, it has antioxidants. But, I mean, just with normal metabolism, we make oxidants. And as we start to age, those oxidants are going to start to do destruction on our cells. One other thing I want to say that's an intermediate. So this is an intermediate, and another intermediate is this thing called 1,3-biphosphogylycerate. All right? So that sounds somewhat familiar, that intermediate product. For those of you who are taking AMP2, don't necessarily expect people not taking AMP2 to know what the heck I'm talking about there. But for those that are, does this sound familiar, but not quite right? Yes, no, maybe? Okay, all right, let me tell you about it. This is normal. This is an intermediate in glycolysis before we get to pyruvate. Now, before I start to talk about that, because you can shift from this pathway to making ATP to another pathway that's going to be important for something else, all right? But let's first just talk about this lovely graph here, where you have partial pressure of oxygen, and here you have saturation of oxygen with you know, a hemoglobin molecule, or the hemoglobin. When they say this, they really mean overall saturation, okay? So this is what's called the dissociation curve for oxygen. It's essentially saying, hey, hemoglobin is going to want to bind to oxygen, and then in certain situations, it's going to want to not bind to oxygen.
Due to like pressure gradients?
Pressure is going to
Partial
play a role with it as well. So if we're thinking about, you know, here we'll have something like 100 millimeters of mercury with oxygen, and then here we'll have something like 40 millimeters of mercury, and then we're going to have this curve that kind of goes like this. So at 100, it's about 98% saturated, the hemoglobin with oxygen. Okay? Everybody good with that? And then like at 40, it's going to be more like 70% saturated with hemoglobin. Where do you think in this curve, where are we at the lungs versus at the tissues? Just thinking of it intuitively. Where would hemoglobin be binding with oxygen? Almost 100% of it is binding to
Probably
oxygen.
the lungs.
Lungs. So in lungs, the partial pressure of oxygen is 100. It's higher there, okay? And so this is going to be the lungs. Now, here, you're seeing that hemoglobin want to release oxygen. And it's releasing oxygen to tissues in the systemic circuit. So the actual partial pressure of oxygen in the systemic circuit is about 40 okay? So this is at the tissues where you're having release of oxygen. Here, you're having oxygen bind to hemoglobin. All right, so that's just that curve showing how, hey, at a higher pressure of oxygen, partial pressure of oxygen, hemoglobin is going to want to fully bind with this oxygen. Where at a lower partial pressure, it's going to want to kick it off. It doesn't have as high of affinity. I mean, thinking about high altitude, your partial pressure of oxygen is lower at a high altitude, so you're not going to saturate your hemoglobin as much. So you're just going to, in general, have hypoxia because hemoglobin isn't going to want to bind to oxygen because the partial pressure is lower at a high altitude. You're good with that? Okay, now, there is this thing that is called the Bohr effect. The Bohr effect is where you get a right shift of this curve. And so you're going to see this happen. All right, something like that. And so now, I mean, this is not to scale, all right? But you should be seeing the general pattern here. When we get to the tissues now, the saturation is 40% saturation at the tissues because we've had this shift. Is that going to be when the tissues are going to have a higher temperature? Does that make sense? Why would they have a higher temperature? What would make a tissue have a higher temperature? Exercise, yes. So we're moving. You know, when you move, you're going to increase the temperature at that tissue as that muscle, for example, is contracting. So we're going to have a higher temperature with this Bohr shift, or this right shift. Now, if the tissues are more metabolically active, are we going to see more carbon dioxide in that area? Yes, because carbon dioxide is a byproduct of cellular respiration. They're picking up more oxygen, making more ATP, and they're dumping out carbon dioxide. Right shift is going to also happen when you have more partial pressure of carbon dioxide. Now, carbon dioxide, when it gets released from the tissues, it's going to bind with what? To make carbonic acid.
Hydrogen.
Which then, this is an acid. Acids like to donate protons, right? This is going to really quickly go into donating that proton and making bicarbonate, right? But what is it combining with here to get here? What? Water, right? Because there's lots of water all around these cells. So, as you increase your partial pressure of carbon dioxide, it's going to quickly go through this chemical reaction. What happens to the pH? Does it go low? Does it get lower or does it get higher? Okay, let's think about pH.
Lower because
First of all, let's think of it this way. Is it going to be more acidic, the environment, or more basic?
more acidic.
More acidic. So let's write that down. It's going to be more acidic. And if we talk about pH, a more acidic environment, is that a low pH or a high pH? Low pH, right? But the whole thing that we measure for pH is hydrogen ions. So we're going to get an increase in these hydrogen ions with this right shift. Everybody good with that? So all these things are happening with tissue that is metabolically active. Now, so we get this right shift. This is just showing that at the tissues you're releasing more oxygen. Everybody good with that? Release of even more oxygen. So the affinity of that hemoglobin molecule to bind to oxygen has decreased. Now back to this. 1,3-5-phosphate glyceride. When you have an acidic environment, this pathway turns into what's called the Roux-Port bearing pathway. And instead of this being made, you make what's called 2,3-5-phosphate glycerate. Also known as 2,3-BPG. Does that sound familiar to you? This particular molecule, because now we've shifted, these erythrocytes aren't making ATP, really now. They're wanting to unload this oxygen to this metabolically active tissue, and now this pathway is doing this instead to make this, which actually also is a part of this right shift. 2,3-5-phosphoglycerate makes hemoglobin unload oxygen. That's the whole function. And so that's why when you get a decrease in that molecule, or I mean increase in this molecule, you increase the unloading of oxygen. Makes sense. Everybody okay with that? Any questions? Yes, Jessica.
So the, wait, no, never mind.
What is your question?
Well, I was.
Normally,
What is the 1,3-5-phosphoglycerate? That is normal, or is that part of the Bohr effect?
it's not part of the Bohr effect. This is when you're doing glycolysis. This is an intermediate in the pathway to making, it makes pyruvate. If we're talking about going into cellular respiration, but it'll really turn into probably lactic acid of some sorts. But this pathway is being done to make ATP, because even though that mature erythrocyte is made, it's made all its hemoglobin, it has to maintain its structure. In particular, it has to maintain what this sodium pump does. And the sodium pump is active transport, so it needs ATP to make sure that those electrolyte balances are proper. But in addition, this is just going to be an intermediate. Okay, so for example, you would have glucose here, and you have a couple steps, and then you get 1,3-BPG, couple steps, get pyruvate here. But you also are getting ATP, and that's what we're going for by doing that. This is just an intermediate. But then, whenever you get this right shift and you increase the hydrogen ions, it comes in here, and those hydrogen ions actually make it to where you're doing this pathway now, and you're making 2,3-BPG to unload oxygen to that metabolically active tissue. Did that help answer your question?
Yeah.
Cool. Other questions?
So that 2,3-BPG, would that cause a third line to the right of the two lines in the Bohr effect or the dissociation curve?
No,
No?
it's just this curve here, all the stuff I have in purple, these things actually all are gonna be happening in metabolically active tissue. You're gonna get an increase in temperature. You're gonna get an increase in partial pressure of carbon dioxide. You're gonna get this more acidic environment that then shifts this to this pathway, so you get an increase in this. And all of those are gonna cause more unloading of oxygen at the tissues. To some extent, you can kind of think of this as these steps as changing the structure of that hemoglobin, where it doesn't really want to bind with oxygen anymore because it's tweaked its structure a little bit. So it's going to want to pop off that oxygen, which is what you want. Other questions? So I can erase this.
That graph is called the dissociation curve?
Yeah, it's a disassociation curve, like disassociation of oxygen from hemoglobin. Like here, it's at 0. Where here, it's at 100%. When it's 100%, that means that most all the hemoglobin is bound to oxygen. Down here, that shows that most, like all of the hemoglobin has unloaded its oxygen. I mean, for the most part, hemoglobin doesn't unload all of its oxygen. That's why for CPR, they don't do breaths anymore, because hemoglobin already has oxygen still bound to it, and they just want to pump that blood through to unload that oxygen to tissues.
I just took my ALS course and we still did rescue breathing.
Yeah.
Yeah,
The
I mean,
compressions are just the most important part because you want to keep that circulation of blood within the body.
right, and I mean, at some point, it will, I imagine it'll eventually unload all of its oxygen or won't want to give it up, that you'll need to start to give oxygen to that person if they're going to survive. It really does make it easier, though, to do CPR not having to do the breaths.
You still have to do the breaths.
Huh?
You still have to do the breaths.
It's just two to.
No, they have a new type of CPR that you don't do breaths. It's just
Compression
compression.
only, if you haven't been
Compression
trained.
CPR.
Okay.
But I mean, like that's for a first responder, like a housewife or a friend. When the second responders come, you know, like the ambulance guy, they always have that thing. Start doing that. You really want to get the heart beating. So there's these cool things around here that can jumpstart the heart. So that's the most important thing, is just to get that heart beating again.
They're called AEDs.
They're called what?
AEDs.
What does that stand for?
I know the D
Automatic.
stands for defibrillator.
Automatic electronic defibrillator.
Automatic electronic
When you
defibrillator.
turn it on, it speaks to you.
Oh yeah, it
walks you
tells
through
you
all the
what
steps.
to do.
Yeah, it tells you what
It's
to
an
do.
external.
Yeah, she's totally right. I was
Yeah.
totally wrong.
I can't remember if it has stuff, because you don't want to put those electrodes on hair. So you actually have
It
to
does.
like shave the hair off of someone.
A lot of them have little razors.
Little razors in it, right? So the hair is just not can't get that contact there. Hairy. Hairy. Okay. Anemias. So that was, the basic stuff. That if you've taken AMP2, you've heard some of this. If you haven't, then you definitely want to go and re-study that. Now, anemias is going to be where we have a deficit in general, a deficit of what? Oh, hemoglobin, which is inside of what?
Red blood cell.
Red blood cell, right? So a deficit, like just in general, anemia is a deficit of red blood cells. And I want to really point out, really what we're talking about here is functional red blood cells, okay? Because you can have red blood cells that don't have the same function as a normal red blood cell. So really a deficit of functional red blood cells. And if you have a deficit of functional red blood cells, that's going to lead to hypoxia. All right, so these go hand in hand. And this is going to be low oxygen carrying capacity. So that's what we mean by anemia. So what was the normal percentage of red blood cells in whole blood? 45%. And that's, average. Men tend to have a higher red blood cell count than females because testosterone, actually that hormone that males make more of, actually triggers red blood cell production, so they have more red blood cells than us. So for them to be anemic, they would have to, be 43% or something, because they can have up to like 45 to 50% red blood cells. Where a female, they don't make as much, so when you start to see like below, like in the 30s for a female, that shows they're anemic. Now, if we're talking about just, in general, manifestations, what two systems are going to be the most compromised or have to adapt to having low oxygen carrying capacity? What two systems in the body are going to change their functioning?
Cardiac and pulmonary.
Yeah. So you're going to see manifestations dealing with the cardiopulmonary system. You're going to have people that have dyspnea, so they're doing like unusual breathing. They might increase their breathing, which is called tachypnea, their breathing rate. In regards to the heart, how do you think the heart would try to compensate with this low oxygen carrying capacity?
Beat harder.
Increased heart rate, right? It's trying to get that blood to where it has to go. So it's going to beat faster. So you get tachycardia going on. But I mean, overall, because so we have these cardiopulmonary things going on. And then in regards to what tissue in the whole entire body is it really important to be able to deliver oxygen to?
The heart, right?
The heart. And so what'll happen here is you're going to get what's called angina pectoris. And this can be transient, so you're not getting enough oxygen to that heart. And so you're going to see that the heart itself, yeah, it's going to start beating faster, but then as it prolongs this anemia, those cells are not getting enough oxygen to make ATP to beat. So they're going to start to weaken, and you're going to get angina pectoris. Then the more that this happens, you're going to eventually get heart failure. So it's like a progressive thing here. Now, just thinking, hey, low oxygen carrying capacity. Now let's think of some manifestations general that could happen with that. Are you going to be really hyper and excited and go get them? Instead, what are you going to be? You're going to be lethargic, fatigued. You're going to be weak. weakness. Overall weakness. Now, depending upon like what type of anemia that you have, you could also be prone to like fainting, you could have headaches. So all of these kinds of things are gonna play into anemia. And it's really about that low oxygen, all right? You're not able to deliver oxygen to those tissues. I am gonna write down on the board, because there is a lot of them that we're gonna go over. And I started, again, just like with, did you guys like that white blood cell malignancy table I did? Did you like that? Okay. I started another table for these, because there's a bunch of them. And so when I have it all polished and nice, I'll put it on CNN Learn for you guys. But we're gonna have, first, it's this thing called aplastic anemia. This one is where bone marrow has been destroyed. Is destroyed or injured. I'll put injured. So that's what the aplastic stuff is. You can also have anemia that is in relationship to chronic renal failure. Now, what is it that the kidney makes that is important for making red blood cells? That's dealing with blood pressure. Let's think, what is triggering the production of erythropoiesis? EPO, which is called what? Erythropoietin. And what organ makes erythropoietin? Kidney. So, when you have chronic renal failure, you get a decrease in erythropoietin. So then you're not going to have this hormone going in the blood to the bone marrow to trigger the production of red blood cells. You get a decrease in red blood cells then. So that's one way, but the other thing that chronic renal failure does is the kidneys have these capillary beds, because what does the urinary system do? Yes, it makes urine, but what is the whole point of the urinary system? To eliminate blood waste. That's the big function of the urinary system, okay? Now, if you're getting chronic renal failure, you're not eliminating that blood waste now. So you get an increase in blood waste. This increase in blood waste actually starts to make it to where blood cells are lysing. It's like a toxin to them. It makes them actually rip apart. So you get blood cell lysing. But then the other thing that's happening in chronic renal failure is that at some point those little capillary beds start to burst. And as they burst, you start to see blood in the urine. And so these people are going to start to just have blood loss that's going to lead to an infection. I mean, this honestly is going to eventually lead to death, but this is hardcore. All right? Now, these two are really about numbers of red blood cells are decreased, right? Straight up numbers of red blood cells are decreased, yeah.
Can somebody have anemia, experience the blood loss through urine, but not be having chronic renal failure?
Usually if you have blood in your urine, there is some type of bursting with those capillary beds that are involved with filtering out blood waste. You can get cysts. People can get cysts on their kidneys. And when you start to develop the progression of making those cysts, You're gonna probably have this, and then it can turn into scar tissue, but eventually, your kidneys, you don't have very much functioning tissue anymore. And I think that it's chronic, so it's a slow progressing type of thing. So first you have these cysts that then turn into scar tissue, and then eventually you have loss of functioning kidney tissue to do what it's supposed to do.
Or people are passing stones too, they can bleed.
Oh, yeah, I could see that, kidney stones, because, and in that situation, it's only localized right there, and so you're gonna burst those capillary beds. But that's just an acute situation that you could probably heal from that, or have enough functioning kidney. Thank you. Anybody else have an example of more of an acute condition? Yeah.
My cat had renal failure. He would drink water all the time, and like, it seemed like he could never absorb water.
Yeah, also with this is when you aren't making, you have a hypothalamus problem. It's called diabetes insipidus, where it's called insipidus because way back when what they would do is they would drink the urine, and diabetes just means an increased outflow of urine. So people who have diabetes pee more. And that urine can either have no taste, which is insipidus, or it could be sweet, which is mellitus. The sweet one is dealing with insulin. The insipidus is dealing with not producing antidiuretic hormone, which allows your kidneys to reabsorb water. Instead, you urinate that water out. So that's probably what was going on with your kid.
That's cool. I remember.
And that's part of this too, actually, hormone imbalances. Now, then we get into ones that, this first one really isn't dietary in regards to its etiology, but it is in regards to not getting proper nutrition. And that, the next one, so these kind of fall in their little category, and now we're getting into this one that's sort of like nutrition. Have you guys heard of pernicious anemia? Maybe you heard of that one. Anyone? Anyone?
Yeah.
What is lacking here?
B12?
Yes, so pernicious anemia is where people can't absorb B12. And really, it's not that they don't have this in their diet, it's that they can't absorb B12. Who knows what is needed to absorb B12? What do you need to be able to absorb B12? It's the only relevant function of the stomach. The stomach makes this, and it's the only thing that you need, that the stomach does that's important for your survival. Everything else the stomach does, you'll be fine.
Intrinsic factor.
Yes! Yay! Okay, so to absorb B12, you need intrinsic factor. And there's these cells in the stomach called parietal cells that make this intrinsic factor. So with people with pernicious anemia, what's happening is it's an autoimmune disease. And this tends to happen usually when you get older, actually. And the body recognizes either the intrinsic factor or the parietal cell as non-self and destroys either of these two things. And so now this person doesn't have this and they're not able to absorb B12. Okay? The other one is folate anemia, which is another type of vitamin B. And this one's just diet. This one, the etiology is just diet. You don't have enough of that in your diet. these two right here are very similar in their manifestations, okay? They're gonna have macrocytic red blood cells, these big cells. They're gonna have, their neutrophils are gonna be hypersegmented. They're gonna have these cells because the DNA, I think it's because the DNA isn't able to synthesize or something, because these guys aren't gonna divide. And that's gonna actually be a consequence of these things that are called megaloblasts. They're gonna have big megaloblasts. If you checked like a blood count thing, you would see these big megaloblastic cells for both of these. The difference that I really think that differentiates the two is what folate and B12 also does for you. So for pernicious, these people are gonna have cognitive dysfunction. And they're gonna also have like delusions. They're gonna have hallucinations. Where this one, these people are gonna be depressed, irritable. They might have a hard time sleeping, which this hard time sleeping might cause these things, honestly. And so that's really how you tell the difference between these two, is based on these more behavioral types of manifestations that you see. This one is actually, because it causes such cognitive dysfunction and these crazy delusions and hallucinations, it's called megaloblastic madness. Pernicious and then we get to iron deficiency. What are the cells going to look like here? Are they going to be big or small, the red blood cells? Small, right? Because you don't have this key component. These are big because this is needed for the cell to divide. These are small because you don't have this key component to make as much functional hemoglobin. So these are going to be macrocytic.
Macro, micro.
I'm sorry, micro. I even wrote micro up here. Microcytic. And hypochromic. I mean, for the most part, all of these are going to have general manifestations. But for iron deficiency, one of the big things that you know if you have iron deficiency is going to be you want to eat ice, you want to chew on rocks. You ever heard of that? People who want, that's called pica. So that'll be a big manifestation with iron deficiency. This desire to eat rocks and ice. Some people even want to eat hair. They just have like because they don't have the proper iron. The other things that you'll see if this is prolonged, some people get spoon-shaped nails. You'll see that. They'll look very pale. A lot of these you'll look pale actually, huh?
I just had a question real quick. The hypochromic, you said the cells look pink, right?
They look really light. Sometimes they might even look white-ish.
But they're a light color.
Yes, they're very light color. They're pale themselves looking, yeah. Now, I'm gonna just, this iron deficiency one, you don't have enough iron in your diet, right? But when we're talking about men, you can get iron deficient anemia if you have, this tends to be one of the things that happens first. Gastrointestinal bleeding. And they'll have like iron deficient anemia because they're bleeding. in their gastrointestinal area. That's more common with men. What do you think is the more common one with women? Increased menstrual bleeding. We can get iron deficient anemia. I don't know how to spell menstrual.
So the gastro,
S-T-U-R-U-R-A-L. Yeah.
Oh, good.
so
Okay, the gastric intestinal bleeding, they're bleeding in their GI tract.
those are a result of iron deficiency or they cause iron?
These are associated, this is an etiology. It's not a manifestation. So, you know, this is a diet one if you don't have enough iron in your diet, but women who bleed out a lot actually end up getting this type of anemia, for example. Or if you're bleeding into your GI tract, you'll get this type of anemia. Because think of it this way, it's essentially blood loss. You're losing red blood cells, but they're normal red blood cells. And so they put it in iron deficient anemia because it's more like just loss of blood that's normal.
So is there hemorrhagic anemia?
Okay, so then you're gonna have, we haven't gotten to this, like we're thinking of these as more intrinsic things, but trauma, like any kind of big, huge trauma. So these, they don't view this as a trauma thing. So if you have a lot of blood loss from trauma, that's called extrinsic blood loss anemia. So you put it in a different category. Everybody still good? I'm going to move over to there then. Are we all cool with that? I just kind of want to keep those general things up there. Then we have these genetic ones. So then you have ones where the iteology is genetics. So those are, kind of diet. The other ones is like aplastic is from some type of injury, some type of physical or chemical injury. Chronic renal failure, you get that anemia, it's almost secondary. So then the genetic ones is you can have this thing called thalassemia. And this is autosomal recessive. If we're talking about an abnormal alpha globin, that is more prominent in Asians. Asians will have this type of anemia. If you have an abnormal beta, this tends to occur in Mediterranean populations. They're just more prone to it. Also with this one, they're having this abnormal globin, whether it's alpha or beta, I think it's more the beta. That means that these individuals are able to, especially if they're heterozygous, they're able to survive malaria. So some of these things are still around because these individuals were able to survive malaria.
Like sickle cell anemia?
Right, exactly. So, and the way that hemoglobin, when it's in the cell, you have this internal cytoskeleton that the hemoglobin kind of sticks to. But if you actually change its shape, it pulls in the cell membrane because it's kind of attached to that cytoskeleton and pulls it in and changes the shape of that cell. And in sickle cell, it squashes that protozoa and kills it when it's in the cell. And so similar things that are messing with these globins and allowing the cell to kind of
So
invaginate
it's
or
like
pull in.
affecting like the spectrum?
There is another hereditary one that's specific to the actual spectrin, that protein, and that's called spherocytosis. And so that's going to compromise the cell membrane in general. This one is the hemoglobin, and since it changes or has a different shape, and it's connected to that cytoskeleton within the cell, it kind of pulls on the cell membrane to kind of squish it in, or sickle. Now, the thing with these guys is, that's interesting, is their hemoglobin, because of having an abnormal alpha or beta, it's not going to bind, or the heme part, let me go with this, the heme part doesn't really bind well to this iron, actually. So you can have a whole bunch of iron that you're bringing into your system, but it's not going to be a part of this heme group. The heme group won't bind to it because this globin is different shaped, and it just doesn't allow for that ring to bind to the iron. All right? So would you think that the cells here would also be microcytic and hypochromic because they aren't binding to iron? Right. So here you're going to have microcytic and hypochromic red blood cells. How would you, now we got iron deficiency, we got thalassemia. These people have enough iron. What ends up happening is that this iron builds up in the liver and spleen. As it starts to build up in the liver and spleen, are those two going to get larger? They do. And so you get hepatomegaly, which is an enlarged liver, and you get splenomegaly. So you'll have this manifestation, but you will not have that in iron deficient anemia. Even though the cells look the same, it's not that you don't have enough iron, it's just that the hemoglobin, the heme, that protoporphyrin is not binding to iron, which is, therefore it can't then bind to oxygen. Sickle cell. Sickle cell is also autosomal recessive. And it's more prominent in the black population. This one is where you're not going to have beta globin. So no beta globin here. Instead, to replace the beta globin, you have, they call it S globin. That's just called, I don't know why it's called S globin. I say it's called S globin because it's sickle. I don't know why they call it that. But I just say, hey, sickle cell anemia, sickle globin. All right? Since these cells go from something like this to this sickle shape, instead, they're going to start to block off blood vessels. And so something that's really common with sickle cell is vascular occlusions. I always want to put a U here. You know, now there's no blood going through that blood vessel. All right? And then you're going to also get, since these are sickling in there, you're going to get in the veins thrombosis. So then you'll have like that vein, its diameter, the lumen gets smaller. And then that thrombosis is going to get knocked off. And as it gets knocked off, it's going to go through the heart, And then it can get into like the arteries, and so now you've displaced it from the veins. And so whenever a thrombosis is displaced, it's called a what? Embolism. So you'll get these arterial embolisms.
So thrombosis is like a blood clot.
Yeah. Like if you have, this is your blood vessel, the lumen, a thrombosis is like plaque that makes this lumen smaller. An embolism is where this guy gets knocked off and starts to travel and goes somewhere else. And by the time and once it gets to a smaller blood vessel, because arteries have a smaller lumen, it gets stuck in there. And then that's where, really where you get these occlusions. And so then you stop delivering blood altogether to the tissues. And you get a lot of tissue hypoxia because of this. Since you're having all these vascular occlusions, you're also just in general having a lot of systems not function properly because they're not getting blood to them at all in sickle cell. I mean, if you're heterozygous for this, your cells aren't going to sickle unless you change something in your environment, like going to a high altitude. They'll sickle. All right.
Thalassemia has sickle-shaped cells also?
Nope. Thalassemia, what do the cells look like in thalassemia? They're small and pale. Right here.
I thought you mentioned that.
Okay. Pale. That's what hypochromic means. These are still genetic ones up here. There's two more. Then I'll let you go. And I'll give you this that has all this. There's some other stuff, and we have polycythemia still to do, but I tend to not give you guys a break, so I might as well let you go early and stuff. Yay. You can go home and pet your puppy and say hello to your husband and hug your kids. Hereditary spherocytosis. This one actually is autosomal dominant. And this is the spectrin one. You have a mutation in the gene that codes for the protein that gives that cell membrane of a red blood cell its structure. All right, so in these guys, since the cell membrane is compromised, it doesn't have that biconcave structure. Instead, it's a sphere. And we've already talked about how that's not going to allow for these people to efficiently deliver oxygen to tissues because you have a smaller surface area for the hemoglobin that's within it. What ends up happening is because that cell membrane is compromised, you have this sphere or red blood cell, these guys are gonna lyse more easily, and that hemoglobin will go out, and the manifestations you see here are like jaundice. You get gallstones. You get, because these are lysing, or essentially they have a shorter lifespan. So as they start to die, where are they going to go?
Spleen.
Spleen. And so you get more of these guys building up in the spleen, so you get splenomegaly from it. Now, thalassemia, sickle cell, and this one. These genetic ones, for the most part, you're gonna have a lower oxygen carrying capacity, which is gonna lead to tissue hypoxia. And so an uncommon, it's not a common manifestation, will be necrosis occurring. Where do you think in the body most of this necrosis would occur? Would it occur up here, or would it occur more in your legs? Legs, right? Because that's a low pressure system, and you're gonna really first start to see these thrombosis going on in the veins. And what ends up happening is, and they don't know for sure, but they hypothesize that these three, one of these uncommon side effects is leg ulcers. And so those ulcers are a consequence of tissue hypoxia, which leads to necrosis and a dying of the tissue. But that is not a common manifestation. The last one is this crazy one that's called glucose-6 dehydrogenase. And for the most part, there's no manifestations. People that have that abnormal, it's an X-linked recessive, the ones that have that anemia, they're missing making this one enzyme in glycolysis, but it doesn't really ultimately affect them. They don't really get manifestations unless they take a certain drug. And when they take a certain drug, they might start to show anemia manifestations. So that one's, okay, that's it. Oh, in regards to the take-home, I'm still thinking, I think what might be fun to maybe get you guys excited, I don't know, would be case studies that I give you to go through and to figure out what type of anemia somebody has, or polycythemia, and I might make that like a take-home type of thing. I'm still thinking about it. I might also make a take-home where it's sort of like a combination of this. The next